Welcome to part 3 of our video on the blood. In the last part, we looked at the different blood cells. We have erythrocytes, which are these biconcave discs. So you see the little indentation, so it sort of looks like this. And they transport oxygen with hemoglobin. In fact, they're just bags of hemoglobin. They lack a nucleus. They lack all organelles. Whereas our white blood cells have nucleuses. In fact, these guys have multiple lobes of their nucleuses. Now, the white blood cells, so these three leukocytes are granulocytes, meaning they have visible granules. The eosinophil right here, it has these brick red granules. Eosinophils are red. And it has a bilobed nucleus. The neutrophil has purple granules. And this one has 4 lobes. 1, 2, 3, 4. And they really are connected. Some lazy people, myself included, sometimes say they have 4 nuclei. It's really 4 lobes. The basophil, look how big it is, actually has a bilobed nucleus as well. But it's obscured by these blue granules. So basophils are blue. Neutrophils are purple. And I love hamburgers. Agranulocytes are the monocyte, which has a large nucleus and some cytoplasm that you can see, and the lymphocyte, which the nucleus makes up most of it. So I only have this thin band right here that's visible. Now our other formed element is this little tiny thrombocyte, which is a platelet. So our leukocytes are complete cells. They're divided into granular, which have visible granules, and agranular. The 3 granulars are basophils, which make histamine, eosinophils, which attack worms, and the neutrophils, site-attacking bacteria. Now, our 2 agranular ones are the lymphocyte and the monocyte. The lymphocyte gives you specific immunity, and it helps you fight off an infection the second time you encounter it. So again, with the coronavirus, people do not have lymphocytes for it because they haven't been exposed to it. Once they get exposed, they develop lymphocytes to attack it when they get exposed again. Now, monocytes actually leave circulation very early on and become macrophages. These are garbage disposal units. They eat up fragments, such as viral fragments, and they play a special role in specific immunity too, but we will talk about that later. So our macrophages, where we left off, are the cleanup units. They don't attack things specifically. They find viral fragments and bacterial fragments, and they actually go and tell the lymphocytes who they encountered. So they're actually part of specific immunity in that regards too. Now, thrombocytes are little tiny platelets, and they're fragments of cells. So they're these little tiny things, probably like this on the actual histology slide. And when they encounter damaged tissue, they become 80s punk rockers. And they help with blood clotting. They form the platelet plug. And you will learn all about this in physiology. So what is Rolex? It's not a Swiss watch. That's Rolex. White blood cells, which are leukocytes leaving venules? No, I don't think so. Red blood cells, erythrocytes, lining up single file to go through capillaries. Yes! And our platelets are thrombocytes. So that's not it. So, tell me the functions of these cells. You need to know what the function is for the exam. So, lymphocytes deal with specific immunity. And they're actually more effective against bacteria, which are in the blood, than viral fragments. So, notice there's no shot for HIV, even though it's been around since 82, 83, because they just can't find it because it hides out in your helper T cells. So it's very hard to make vaccines for some viruses. Neutrophils, what do? They're phagocytes. They eat bacteria. They're very good at eating bacteria. Viruses, again, hide out in cells, so they could eat the capsids from the virus when it's out, but it's very hard when the virus is not hiding out. And eosinophils, what do? They attack worms. Good. We're now going to look at how they develop, and how they get recycled for red blood cells. We're going to look at which are myeloid, which are lymphoid. And you've heard of lymphoid leukemia and myeloid leukemia? Well, those are from those cell lines. And we'll talk about the life cycle of the red blood cell. So, all blood cells start out life in the bone marrow from this thing called a hemocytoblast, which is a stem cell. Now, early on, that stem cell will have two fates. It will become a myeloid or a lymphoid stem cell. Now, all of the lymphoid line become lymphocytes. There's debate whether or not they become this thing called a lymphoid dendritic cell and a mast cell. We're not going to worry about. So lymphoid cells become lymphocytes. So the myeloid line, it makes everything else. The basophils, the neutrophils, the eosinophils, the monocytes, the erythrocytes, and the thrombocytes. So myeloids make everything else. So this question isn't that hard in the depth of field I'm going over. For the stem cells, the lymphoid stem cells make all lymphocytes. The myeloid stem cells make everything else. So thrombopoiesis is the production of thrombocytes. So thrombocyte, poiesis making. They start out life as a megakaryocyte. So this is a megakaryocyte. It's actually a really cool cell. It has anywhere from 7 to 12 nuclei. I think the number usually listed is 9, though. So it has this multi-nucleated cell. And this cell is busy making ribosomes, which make proteins, and RNA, which code for making proteins. And the platelets are going to break off as little tiny fragments. These little tiny fragments breaking off, and they're going to circulate. And they could circulate for a long period of time. Now, they play a role in blood clotting. They may have some regulatory and immune factors as well. It is not completely sure. So, megakaryocytes give rise to platelets, and it's a multinucleated cell. Now, all lymphocytes develop from the lymphoid line. Now, the B cells are bone marrow, and the T cells are going to mature in the thymus. All other cells are myeloid. So here's our lymphoid line right here. B cells, bone marrow, T cells, thymus. All others mature in the bone marrow, and it makes all other cells. The granulocytes, the agranulocytes. So, red blood cell production is controlled by a hormone called erythropoietin. Now, erythropoietin controls how fast the red blood cells divide. And it is triggered by low blood oxygen levels. So we have low oxygen. That triggers EPO to be released. Now, this is the hormone Lance Armstrong abused, by the way. EPO goes to the bone marrow, and it increases red blood cell production. And more oxygen is carried, and it returns to normal blood oxygen levels. So I went to Die Matterhorn, and there's an observation deck on Der Klein Matterhorn. Don't ask me why one is D, one is Der, by the way. Now, at Der Klein Matterhorn, the elevation was 4,000 something meters. There were oxygen tanks everywhere because of low oxygen levels. There were elderly tourists sitting with the oxygen tanks because they had low oxygen levels. Then there were these two hikers, Hans and Franz. That might not be their real names, by the way. Now, they were going off, and they were going to hike between Der Klein Matterhorn and Die Matterhorn. They had no problem hiking across the glacier. The reason they had been up at that elevation in Zermatt for about a month, and they had acclimated because they made erythropoietin, and the red blood cell number was greater. When I train in the summertime, I love it when we go up to the mountains because I'm running and biking at elevation. The first bike ride and the first run suck, maybe the second and third one, but after that, it gets easier because I'm making more red blood cells. And this takes about a week, and yes, we go to the mountains for more than a week. Now, a brief overview. Erythropoietin, called EPO, causes myeloid cells to form pro-erythroblasts. Basically, blast is a building cell. So we have our pro-erythroblast. Then it goes through two boring divisions. Now, since these cells are dividing, they still have a nucleus. So here's our pro-erythroblast, and it has a nucleus. It does its division, still nucleus. So this thing that's left over still has its nucleus. Can you see it? Now, this thing is called a reticulocyte. So over here, we have our stem cell, our pro-erythroblast, our two boring divisions. And then we have our reticulocyte. That still has a nucleus. The reticulocyte is actually released into circulation, so this picture is wrong. And it eventually is going to eject what's left of its nucleic acids. So this has nucleic acids, and they stain. So you can see them in some of the bloodstains. Hanging out with this neutrophil, we have a reticulocyte. We have some nucleic acids right there. That might be a reticulocyte too. They tend to be less than 3% of circulating red blood cells. So after 24 hours, they lose their nucleic acids and they become a normal erythrocyte. So a reticulocyte, is an immature red blood cell in circulation. It still has these fragments of nucleic acids, typically 1%. Higher than 3%, 2.5 I rounded, it means you have increased red blood cell production. It could be a sign of something called hemorrhagic anemia, where you have red blood cells breaking. You're bleeding internally. It could be anemia where your red blood cells are just breaking down at a humongous production level. Or it could be you're up at elevation, your favorite Swiss Alp, and you're just making more because you just got there. So a reticulocyte is just an indirect way of saying red blood cell count increased. Or you're Lance Armstrong and you're taking EPO illegally. Now, the mature red blood cell lacks all organelles. They can't divide. They can't repair themselves. So they get beat up. They're very small cells. They're flexible. But this right here is going to beat up the red blood cell. And they get worn out in about 90 to 120 days. So, unable to divide, grow, or synthesize proteins, they wear out. The date I want you to know for the test is 100 to 120 days. And they get eliminated by the spleen. Now, one of my students, this was years ago, she had a disorder where she had large circular red blood cells. They were getting beat up faster than normal, so they were living maybe 50 days. She was having all sorts of problems with her spleen and with her liver because of this. And she actually had her spleen removed because her red blood cell recycling was way too fast. By the way, the liver also helps recycle them. So, in the spleen, the surface is checked. If it's irregular, if it's rough, it stands a chance of triggering a platelet, and the platelet will cause a block in a blood vessel. So it gets destroyed by a macrophage. They really are big eaters. The iron is going to be recycled. It gets sent back to the liver. Most of the hemoglobin is recycled. Problem is this heme group in the center, right here. The heme group has all these rings. The body can't handle rings. So it gets sent to the liver, where it becomes bilirubin, and it goes to the kidneys to be excreted. Now, the reason why your urine is yellow is because of bilirubin. Now, some of it's excreted by the liver in the form of bile, and the bacteria in your intestines love it. They eat it, and it actually turns your fecal matter brown. The last slide tells that what organs do what in hemopoiesis, and the kidneys detect low oxygen level and make EPO, and they dump bilirubin. The liver makes a hormone for platelet production and recycles erythrocytes. The spleen recycles red blood cells also. Bone marrow makes all the myeloid cells, and the thymus develops the T cells. And I'm sorry, but we don't have time for the questions at the end of the PowerPoint. Have a good day.